All right, let's cover the second half of photosynthesis. And this is what we can call the dark reactions. That's kind of an old fashioned name for it because it's something that could take place even if you took a plant out of light. These are just chemical reactions that are going to be driven by the energy that's been stored during photosynthesis. So, yeah, the interesting thing about photosynthesis, well, another one, I guess I keep saying the interesting thing, but the latest interesting thing about photosynthesis is that it actually does not directly generate very much energy. So you get a little bit of NADPH coming out, which is an electron carrier, and you get a little bit of ATP that's made, which is driven by the hydrogen. We call these just protons. Since hydrogen without an electron, H plus, is just a proton floating around, which may or may not have a neutron attached to it, but usually doesn't. So we'll sometimes call these like a proton gradient or a hydrogen gradient. And so we're going to build that up on the inside here because this is where we are splitting the water to make oxygen and H plus, hydrogen ion. And hydrogen positive ion is then going to feed through this molecule, which does look like a giant lollipop. I've drawn it upside down, so I guess it looks like a thermometer. But yeah, the top actually spins around. It's a beautiful big molecule. And if you look, there's a video that actually covers what this thing looks like, a very nice molecular animation of the ATP synthase. So when you see the big thing on a stalk that is rotating around and smashing molecules together, that is the ATP synthase, and it's making ATP from ADP, adenosine diphosphate, plus the PI is phosphate inorganic, which just means phosphate, not really attached to anything. All right, but what we are covering in this half is going to be the Calvin cycle or the dark reactions, and so it's essentially all this stuff over here in the circle of arrows at the right side. And we're going to need to know what some of the products are and basically how this thing keeps moving. This circle, the Calvin cycle, is what is actually going to generate most of the ATP for the plant cell. So it is only doing photosynthesis, so it can have just barely enough energy to do a much more difficult task, which is to strip CO2 and get rid of two oxygens that are each not even single bonded, but double bonded to that carbon, and to pry each of those off, basically buy them off with free electrons. And with that, we are able to then use the carbon and join 6 of those carbons together and make a sugar molecule. It is a ton of work, and thank goodness plants do it because this is where most of the energy that drives the entire biosphere comes from. This photosynthetic reaction in some form or other. All right, now a chloroplast is actually a lot like a mitochondrion. We think they were both originally bacteria, and if you look through the videos, I have a video that'll take you through pictures of some very mitochondrion-like bacteria that exist in the world and still have a lot of the same proteins. Genetically and structurally, they're very close. They're in a group of bacteria that we call the rickettsia. And then you've got chloroplasts, which look very, very much like a cyanobacterial cell, which is a kind of bacterium. They're sometimes called blue-green algae, but really algae are all eukaryotes. And this is a prokaryote, so we shouldn't call it that. That's just weird. Cyanobacteria is a much better and more scientifically correct name. And we've got a nice little thing that shows you the structure of a chloroplast, and also a thing that shows you the structure of cyanobacteria. So you can compare those. So you can compare mitochondria with mitochondria-like bacteria, chloroplast with the chloroplast-like bacteria, and this should convince you that, wow, there's a lot of similarity here. And I'll tell you, you're just looking at the similarity in morphology and the way things look. But the similarity does extend right down to proteins and to DNA. So yeah, the similarity runs deep, and that's why we think that this is the origin of mitochondria and chloroplasts. They're basically bacteria that live inside of our cells permanently now. Okay, so what else do these two things have in common? Well, they're both generating energy in different ways. So a chloroplast is using this relatively inefficient solar power, and by doing this, it is making H plus, and it's making the H plus by splitting water molecules into the oxygen, which then it joins up to make oxygen gas O2, and the hydrogen H plus, which is then going to power the ATP synthase. And actually, in both of these cases, the chloroplast and the mitochondrion, the goal is to generate a hydrogen gradient, a proton gradient. So there's a lot more H plus on one side of the membrane than the other. And this H plus is going to be building up on one side, and it's going to be trying to get through to the other side because of a thing, I don't know, you might have learned as Le Chatelier's principle, which is just to say that if you have a whole lot of something on one side of a semi-permeable membrane and not so much on the other side, the molecules will move and rearrange themselves until they come to an equilibrium. And they'll basically try to equilibrate, like equalize, the concentration of ion on both sides. And that's exactly what's happening here, but the H plus can't get through the membrane on its own, but we put this nice little ATP synthase in there that will load H plus on one at a time and use that to actually physically spin the molecule around. And as it spins around, it will physically crash ADP and inorganic phosphate together to make ATP. Yeah, it's just physically punching two things together until they bond, which is also a way that chemistry can happen. Yeah. Yeah. All right. So mitochondria and chloroplasts both have these ATP synthases, and they are both driven by H plus. So yeah, there's a lot in common. And remember that plants have both chloroplasts and mitochondria. So the chloroplasts are going to store up energy that's used to make sugar, and the mitochondria in a plant, just like in a human, are going to burn that energy, burn that sugar, in order to get ATP. So yeah, plants just go one extra step. Okay, so we're going to talk about the Calvin cycle. Here we go. It's going to be great. And so just the basic overview. This would not be enough for a test question answer, but I want to get you started gently. So carbon is going to enter into the cycle, and we're going to follow the carbon around rather than diagram out the exact shape of all the molecules, which is something that is known, but I don't see a whole lot of value in learning that. We learned the protease mechanism right down to molecular detail. I feel like that's enough for this particular one. But yeah, it's the next step. It's where you could go if you end up specializing in Calvin cycle or plant biology later on. Okay, so carbon dioxide, CO2, comes in, and eventually carbon is going to leave this process as glyceraldehyde 3-phosphate. Yeah, and so glyceraldehyde 3-phosphate actually has 3 carbons on it. And so in the process of doing this, we're going to take a 5-carbon sugar, add this carbon to it to make 6 carbons, then split that into 2 3-carbon sugars, one of which will leave the cycle, and the other of which will join up with something else and eventually go on, and we'll get back there to where we can add another CO2. I've left some gaps in there. We're going to fill those in on the next slide. So for the net synthesis, like overall, we have to actually, of one glyceraldehyde 3-phosphate, G3P we can call it, we are going to have to fix 3 molecules of CO2. So we're going to have to actually go around 3 times. Yeah. And yes, you could divide up the Calvin cycle into 3 phases. So carbon fixation. Fixation means taking CO2 and turning it into something that is a little bit more useful. So knocking at least one of the double bonds off of CO2 and attaching it to something else. That's the idea of carbon fixation. And on the planet, there are bacteria that can do carbon fixation, and there are also plants that are very good at it. So think of it in terms of if we wanted to colonize somewhere like Mars or the Moon. There are a lot of elements there on these planets that we could potentially turn into soil, but what we're lacking on both of those planets is free fixed carbon. So before we could actually just move and start farming on the surface or maybe inside of a dome like in The Martian, on Mars, let's say, we would need to we would want to send ahead some kind of bacteria or maybe a lichen or something like that. They're pretty tough to actually do some carbon fixation. This would be the process called terraforming. And that's not on the test. That's just interesting stuff that you can have in your life. Okay, so the second phase is going to be reduction, where we actually are going to pry the oxygens loose from that carbon. And then the last step is going to be regeneration of the CO2 acceptor, which is ribulose bisphosphate. And the carbon fixation is done by a very special enzyme called rubisco, which is ribulose or ribulose bisphosphate carboxylase. And I'll show you that written out a little later. And we actually have a link in the Blackboard site that will take you to the rubisco molecule. And so you can rotate it around, see what it looks like right there on the screen. It's kind of neat. Once you click that link, it'll take you to a page and it'll show you a protein model of rubisco. And if you just click with your mouse or with your finger on a cell phone, I think, on any part of it and sort of move it around, you'll see you can rotate it. You can zoom in by sort of stretching it out, or you can zoom out by pinching. You can go inside the molecule. Yeah, whatever. Just a neat little tool. Okay, here is our carbon fixation diagram. And this is going to be enough to actually pass that test question. This plus a little bit of knowledge from the end of this lecture. So, like I said, we're going to follow the carbons. And so the carbons are going to be little gray circles in all of these. And we're not going to be paying attention to what's bound to the carbon, whether there's an oxygen here or a hydrogen there, something like that. All of these molecules have other atoms bound to them. We're just following the carbon here to make it a little bit simpler, and just so we can see how the process works, because that's all I'm trying to get out of this particular one. Okay, so at the start, you're up here, and you're going to have 3 of these molecules of ribulose bisphosphate, which is RUBP. And so ribulose is going to be the 5-carbon sugar here, 1, 2, 3, 4, 5. And bisphosphate, the bis means twice. It's a French word. Apparently, if they want an encore of something in France, they don't always say encore, even though that's also a French word. They'll sometimes say bis, which means do it again. Yeah, I liked it. Do it again, twice. And so this is ribulose, sugar in the middle, with twice phosphate, 1 phosphate on either end of it. And the phosphates are going to be important. They're going to be involved in, you can think of them as being involved in something like a tug of war. Because the phosphate molecule is a phosphorus surrounded by 4 oxygen molecules. And those oxygen molecules are all going to be pulling on electrons. They're going to be straining bonds, just taking up way more electron time than they should be, than a normal atom like carbon would. Okay, so we've got this, and the next step is that the enzyme rubisco, ribulose bisphosphate carboxylase, that's where the name comes from, is going to fix 3 carbon molecules. You're going to fix 1 onto each one of these things. So you started out with 3 of these, you're going to add 3 carbons, and you're going to add them right here on the second position. And let's do that because that gives us a nice visual picture, and it gives you something to kind of a hook that you can hang the idea on in your brain. And so now we have a molecule with 6 carbons and a phosphate at either end. Now, this is very unstable, does not last for long, and there are enzymes involved at just about every step of this process. Like over here, where it has multiple arrows, there are enzymes involved, there are intermediate products involved, and we are not going to cover them in that level of detail. So for what you need for the test, it's basically what's on here, plus a little bit from the end. Okay, and this 6-carbon thing, these 3 6-carbon things rather, are going to split, and keeping the numbers straight really helps with the math here. So we got 3 6-carbon things. We got a total of 18 carbons, and they're going to split right here. So imagine those 2 phosphates being like ends of a tug of war, and they both pulled on the rope so hard that it snapped right there. And so we're going to make 3 of these molecules that have a phosphate on one end and 3 carbons. And now you see the carbons are in an L shape here, but in a line shape here. Neither one of those is exactly correct. Carbons will just bond and they can actually rotate around to some extent. I believe you're actually going to literally get two of the same molecule out of this reaction. So just imagine that third carbon is rotating down where this one is, right there. And then you got it. But 6 3-carbon sugars is still 18 carbons. And so we haven't lost anything, we haven't gained anything. This is good. We're conserving. And our math still makes sense. We're still able to balance our checkbook, in other words. So we need to know the structure of it, like where the carbons are, where the phosphates are, and we need to know the names for all the things that have names in here. So this is 3-phosphoglycerate. So the name for a 3-carbon sugar is going to be glycerol or glyceraldehyde. If it has glycer at the front, that means it's a 3-carbon sugar of some sort. And so glycerate, fine. And what this is saying is that on the third carbon, 1, 2, 3, there is a phosphate attached. So third carbon has a phosphate attached to glycerate. That's the name of the molecule. And if you know the name, that tells you exactly what to draw. Or if you know what to draw, you should be able to figure out the name. Works both ways. Chemistry is nice like that sometimes. Okay, so now we have 6 of these. And so the next enzyme in this process, and we're not going to memorize the name of the enzyme, but the next enzyme is going to take 6 ATPs left over from actual photosynthesis, and it's going to smash them. This is a disaster for the plant. It's just lost some energy. And so it's made 6 ADPs out of the 6 ATPs, but it's taken that third phosphate. Remember, ATP is adenosine with 1, 2, 3 phosphates. Adenosine 3 phosphates, adenosine triphosphate. And we've changed it to adenosine diphosphate, and diphosphate means 2 phosphates. So where did the extra phosphate go? Why, we joined it on to the other end. And so now look at this name, because the name makes a lot of sense. So this was 3-phosphoglycerate. This is a molecule with a phosphate at both the first carbon and the third carbon. And so what else can we call this? We don't really have a choice. It is 1 for this one and 3 for this one, phosphoglycerate. Bisphosphoglycerate, rather, because it's got 2 phosphates on it. Yeah. So 1 and 3, bisphosphoglycerate. That's the next molecule in our chain. And if you know the name, you know the molecule, and vice versa. And how many carbons do we have? Let's do our little carbon math. 6 times 1, 2, 3. We're still at 18 carbons. Nothing gained, nothing lost. All right, you remember that the light reactions of photosynthesis produced 2 products for us. One was a little bit of ATP, and we've just smashed that and wasted it, so great. The other thing it produces is a little bit of NADPH. And this is a substance that we made. You would have seen it as NADP. I guess that's a related substance. This has an extra hydrogen on it. But this NADPH is going to be smashed once again. This is an electron carrier. And so we're going to get an electron out to feed into the system. And we're using the electron to make one of these phosphates leave. All right. And so it looks like we have made the phosphate leave that we just put on, but it's actually the other one. I think this molecule ends up flipped around backwards. But all we got to know is that it's 3 carbons long, so it's going to have glycer in the name. And this one's actually a glyceraldehyde. And at the 1, 2, 3, third carbon, and chemists have very specific ways about how they name these carbons. So they've shown it kind of flipped over so that the phosphate is still on the third carbon as you would count them out. And so that's how it makes sense. And so this is glyceraldehyde 3-phosphate. This is a very important molecule because we have a couple options here. One, we can just spin our glyceraldehyde 3-phosphates around, and 5 out of every 6 are going to go right back around the circle. 2, second option is we can take these glyceraldehyde 3-phosphates out and we can do one of 2 things with them. We can either stick 2 of these 3-carbon sugars together to make glucose, which is a 6-carbon sugar, and that's fantastic. We have just stored a glucose molecule that we can then break down and get many copies of ATP out of, much more than we invested in making it. Or we can take this molecule and we can just use it for something. So this can actually directly enter the second half of the glucose breakdown process called glycolysis, Or, yeah, we actually build a lot of amino acids, even some fat molecules, out of these little glyceraldehyde 3-phosphate molecules. So they're very useful. Okay, so here we have 6 times 3 is 18 carbons. So we're going to take one of these out. And, okay, so that's 3 carbons missing. So we got 3 carbons here. So we need 15 carbons, because that's what's going to be left. And sure enough, there's 5 of these glyceraldehyde 3-phosphates that are going to go over here. And so we've just sort of pumped one of these out to a storage container. We're pumping out about 1/5. It's not exactly 1 out of every 6, that went around. It's just that when you build up to a certain level, it takes, it takes 6 of these to drive the cycle, and you're going to subtract 1 roughly for every time you drive the cycle. All right, so we've got 15, 16, 17, 18 total carbons. 3 of them are going to storage. We're not going to bring those back. And so now we've got 15 carbons up here, and we've got 5 3-carbon sugars. Now, if you were to follow through the enzymes involved in this step, and this step, they would do some wonderful things. And what they're going to do is join these together in such a way that 5 3-carbon sugars are going to make 3 5-carbon sugars. And just to get us ready for the next step, because ribulose bisphosphate had 2 phosphates on it, hence the bisphosphate, we're going to have to smash 3 more ATPs over the ends of these things to add that second phosphate on. So we'll keep the phosphate down here, right there, and we'll add a second phosphate on. And now we have regenerated 3 ribulose bisphosphate molecules. And so these 3 can then have another 3 carbon dioxides added. They'll join right here and go around to 3-phosphoglycerate, which will then have a phosphate added to the other end of it, bam, which will then have an electron dumped into it so that we can get one of those phosphates, the one that was already there, to leave. We'll flip the molecule around backwards, and we're going to call that glyceraldehyde 3-phosphate. One of those will go off for storage. The other 5 are going to go around, and through the magic of biochemistry, we're going to change 5 3-carbon glyceraldehyde 3-phosphates into 3 5-carbon ribulose bisphosphates. It's still 15 carbons, and we're still going to add 3 more to get back up to 18. That's the cycle. That is the Calvin cycle. That is the detail in which you're going to need to learn it for my test. Here's the Calvin cycle in a little bit more detail. So like I was saying, there are names for every intermediate involved in this process. And there are enzymes that catalyze every one of these reactions. So, yeah, there's a lot more here. There's a lot more depth here that we could be learning. But I would say for the purpose of this class, we are not going to need to learn this. But if you're curious, if you want to know, well, how did something happen? What's going on with the 3 or 4 arrows over on this side? This little thing will actually tell you. All right, so that's the basics of the Calvin cycle, and now to finish out our talk about carbon fixation in the Calvin cycle, we need to talk about a couple other small topics. All right, so in some conditions, Rubisco will add an oxygen instead of a CO2, and this is a complete disaster. Let's go back 1 slide. Let's see what would have happened. Actually, let's go back 2 slides. So here we got 5-carbon sugar, and then we're adding a 6th carbon onto each one of these 3. And so we're making 3 6-carbon sugars. It's very nice. But if we instead added just an oxygen, we're not adding any carbon here. And so we just have a 5-carbon sugar, but we have an oxygen on it. And it turns out that the only way to get that oxygen off is to go through the entire process. So normally when we go through this process, we're going to burn 6 ATP here, 6 NADPH here, and 3 ATP over here. But we're going to get out a glyceraldehyde 3-phosphate sugar that we can actually turn into all this energy and more later on. So net positive. But if we put in an oxygen, we still have to spend 6 ATP, 6 NADPH, and 3 ADP. We don't get any extra glyceraldehyde 3-phosphate sugar out here. We just take our existing 5 around here, and they go around the cycle, and they start over again. So we waste material and get nothing for it. That's bad. And this process is called photorespiration. When Rubisco, ribulose bisphosphate, adds an oxygen molecule instead of a CO2. It turns out that Rubisco despite being the foundation for essentially all of biology as you and I know it, is kind of inefficient. And so plant cells are actually, they have these areas that are just absolutely stocked, totally chock full of Rubisco enzymes, because each one is not particularly efficient. And the plant needs to make sure that it keeps generating energy, basically, with all the products of its light reactions of photosynthesis. And so this is an area where synthetic biology, which is another branch of biology, can potentially come into play. In synthetic biology, we would be taking existing natural things and trying to change them, trying to make them work a little bit better. And we have some pretty powerful tools that can actually do this. It's a very interesting area of biology. And yeah, there's a lot of money to be made in this area, I would say. Okay, anyway, so that's photorespiration. That's why it's bad. Now, most plants are going to be kind of plant called a C3 plant, which means they're going to make a 3-carbon compound with their brand new CO2 that they've joined in, which is that 3-phosphoglycerate. That's the initial 3-carbon compound that they're going to make. And so that's fine. Let's see, in photorespiration, yeah, we produce a 2-carbon compound. There are a couple of enzymes that are going to work on this and get us back around, but we essentially have to pay the full cost in terms of ATP and NADPH without gaining any sugar at the end. All right, so C3 plants have this problem. Just occasionally they misfire, and Rubisco is going to put in a oxygen molecule instead of a CO2. So enter the C4 plants. And these are just going to take one additional step. So they're going to store their CO2, instead of just storing it in that 3-carbon sugar, they'll actually store it in a 4-carbon sugar, and it's called phosphoenol pyruvate. And so the enzyme that's going to do this is PEP carboxylase, rather. It is going to add a CO2, to phosphoenol pyruvate. And so that is going to fix an additional carbon. This fixed carbon is then going to wait, and or it can be shipped to a different cell that can be a specialist at doing the Calvin cycle. So this is a way to separate the light and dark reactions of photosynthesis. And C4 photosynthesis is, just like it says, much more efficient than C3 photosynthesis. So corn is already C4 photosynthetic. Rice, if I remember right, is mostly C3 photosynthetic. And there are people that are trying to engineer C4 pathways into rice with some success. But you could theoretically do this with a lot of crop plants, and they should just become much more able to store energy. So you should be able to make a plant that's able to produce sweeter fruits or store more starch to feed more people, and stuff like this. Synthetic biology like this is going to be very important in keeping everybody alive as the world population continues to go up. All right, so here's a little bit about what C4 does. You're going to have the regular reactions, but in addition to those reactions, so here's our Calvin cycle as we know all about it. We are going to have PEP carboxylase. This is our special enzyme that does the C4 pathway, and this is all it takes. Phosphoenol pyruvate, which is the PEP here, is actually a molecule that's going to be present in all plant cells, I believe. So that's already going to be there. And if we have this extra enzyme, and it's just one enzyme encoded by one gene, then we can carboxylate that PEP. And so we can take a CO2 molecule, add it on to the phosphoenol pyruvate, and we can store that as a thing called oxaloacetate. And that's then going to get transferred over here, and it's going to go into the next cell. And in the next cell, it'll actually split. And so the CO2 will come loose. We'll have pyruvate that'll then go back in here and get phosphate added to become phosphoenol pyruvate, which is our little 3C compound. And this CO2 will then enter the Calvin cycle once again. And so that's fine. And now that we're down here, we can control. So this cell is going to be away from the surface of the leaf, and so it's not going to have a lot of oxygen. Because remember, the oxygen is produced by the light reactions. And so if you're doing photosynthesis in the same cell that is doing the light reactions, rather if you're doing the Calvin cycle in the same cell that's doing the light reactions, then you will have some oxygen around, and you will occasionally do a little bit of photorespiration, which is very bad. Okay, a different way to avoid the problem of photorespiration is with CAM, which is crassulacean acid metabolism. And so this is another pathway that can limit the amount of photorespiration. Yeah, sorry, I'm struggling with that word today. So this is something that you see in a lot of succulent plants, like there's a bunch of little cactus shown here at the bottom, adorable as they are. There's also things like pineapples that will do CAM metabolism. And so all these plants will have little stomata. This is how they open up to let in CO2 and let out oxygen, which is their waste gas. And they will keep those stomata closed during the day. And so they are just going to generate a bunch of oxygen. And then they're going to get rid of that oxygen and let in the CO2 at night. And they are, in the meantime, able to store CO2 in the form of, there we go, this, CO2 incorporation. So it's going to be another way of fixing carbon. And they're going to crassulacean acid. That's the intermediate that they're going to make. And so they're going to fix CO2 with onto a crassulacean acid, which is this organic acid you see up here. And then they're going to break off the CO2 and feed it into the Calvin cycle, but in a different cell. So it's a way of separating in both time and space the two sides of the Calvin cycle. The carbon fixation is done at a different time and place than the light reactions of photosynthesis. So yeah, that's the idea between C4 and CAM. That's the big difference, which is actually fairly small. And so the test question that you will get will be to tell me about the Calvin cycle and then explain how C4 and CAM plants alter the Calvin cycle. What's the extra step on there? And it's just the one extra enzyme in each case that actually makes them different. So there we go. That's the end of photosynthesis. And now that we have made the energy, we are going to spend that energy. That's the next thing we're going to cover is going to be glycolysis. And it's time to make some ATP. Let's go get that bread, basically. Thanks very much. This has been photosynthesis.